Okay, so you're able to see. I'm sure you're able to see my screen. Can someone just verify that you're able to see my screen?
We're able to see, sir.
Oh, good. Okay. So what we are going to cover is what we call DNA replication. Okay. So what are our objectives? So first of all, we should define what DNA is before we get to the objectives. So I'm sure you did the different biomolecules. I don't know if you've done water already, water, proteins, carbohydrates. You did that already, I'm sure.
Yes.
Oh, okay, excellent. Okay, so you also did nucleic acids, I should think so, nucleic acids.
Yes.
Oh, okay, excellent. So now we are going to look at DNA replication. So first of all, you should understand that DNA is a biomolecule. Okay, so it is an independent molecule. DNA stands for what? Deoxyribonucleic acid. Okay, the fact that DNA stands for deoxyribonucleic acid, the name entails that it is made up of nucleic acids, and the type of sugar that is there is a deoxyribose sugar. When we say deoxyribose sugar, I'm sure you know the difference between a ribose sugar and a deoxyribose sugar. When they say deoxyribose sugar, it is a pentose sugar, to be specific, a ribose sugar that doesn't have an oxygen on carbon 2 in its cyclic form, okay? In that cyclic form of the pentose or the ribose sugar, there is no oxygen. So it is deficient of an oxygen atom at carbon 2. So it is a deoxyribose sugar. And you should take note of something that for you to form a nucleic acid is simply a polymer. You guys understand when I say polymer and monomer, isn't it?
Yes.
Okay, excellent. So nucleic acids are simply polymers of nucleotides. They are simply polymers of nucleotides, and the nucleotides that we have are actually different. And a nucleotide is made up of a nitrogenous base, a sugar, and a phosphate. It is made up of 3 things. So you guys should understand the composition of a nucleotide, that it is made up of 3 components. It is made up of a sugar, a nitrogenous base, a sugar, a nitrogenous base, and a phosphate. So those 3 components will form what we call a nucleotide. Well, a nucleoside is only made up of a sugar and a nitrogenous base, a sugar and a nitrogenous base. So I'm sure you guys did nucleic acid separately. So you're at least experts in terms of understanding those structures, okay? Because today's session, we are interested in DNA replication. So I was just trying to give a brief background on what we are talking about so that it becomes clear even as we proceed with this session, okay? So talking about DNA replication, it is important for you to understand that DNA is found in the nucleus. It is found in the nucleus of both eukaryotic cells and prokaryotic cells. Okay, both eukaryotic cells and prokaryotic cells, you have DNA, okay, which is actually double-stranded DNA. Okay, so it is important to understand okay. Let me share my my camera. Okay. DNA is double-stranded. Okay. It forms a double helix. Okay. Okay. So roughly, this is the double helix. Okay. So we've got the phosphate backbone, okay. Then we've got nitrogenous bases. So now what I want you guys to understand is that there were two guys by the names of Watson and Crick. Watson and Crick. Watson and Crick. So these two guys are the ones that discovered the structure of DNA. Okay. They are the ones that discovered the structure of DNA and understood DNA in quite some detail. Watson and Crick. So what did they do? So talking about the composition of DNA, quite all right, we know that it's a polymer of nucleotides. So we have nitrogenous bases, so and the sugar. So you've got a sugar, then you've got nitrogenous bases, then you've got a phosphate like that. Okay. Remember, the phosphate is forming the backbone. Then you've got the sugar. Then you've got the base. Okay. Now, there are two types of bases that you guys should know. We've got pyrimidines, and purines. Okay. So you've got pyrimidines and purines. So now, how do you know which ones are pyrimidines and which ones are purines? Okay. So let me give you a simple way of understanding or remembering what pyrimidines are. So I'm going to use the mnemonic, cut the pie and pure as gold. Cut the pie, then pure as gold. Pure as gold. Like that. Okay. So what we mean is that when we say cut the pie, we mean that there are three pyrimidines. C is for cytosine. U is for uracil. Then this other T is for thymine. Okay. So cytosine, uracil, and thymine are pyrimidines. These are pyrimidines. So that is how you remember, by simply using the word cut the pie, you are able to figure out which ones are pyrimidines and which ones are purines. So then the pure as gold, the pure stands for purines. Purines like this. Then we've got the as gold here, the A stands for adenine. A stands for adenine. Then the G stands for guanine. Okay. So we have adenine and guanine. So adenine and guanine are the purines. Adenine and guanine are the purines. According to Watson and Crick, understanding the structure of DNA, in terms of their chemical structure, the pyrimidines are smaller in terms of structure-wise. They are smaller, or rather, can I say shorter? Okay. While the purines on the other end are actually longer or larger, okay, in terms of structure. So they are relatively larger structures compared to the pyrimidines. Okay. So what they discovered was, in order to fit in the diameter of DNA, okay, we've got this diameter of DNA. In order to fit in the diameter of DNA, the pairing, we cannot have a situation where you get only pyrimidines, because pyrimidines are short, okay? They will not fit in the diameter, okay? They will not fit. Imagine this one is ending here, and you've got another one ending here. Okay, they are not fitting in the diameter. So which combination should we have? And again, if we use purines, which are very large, they will still not fit in the diameter because it will be so large that the other one won't fit. And you don't want a situation where the two are overlapping, okay? So you want them to fit exactly in the diameter of DNA. So what Watson and Crick discovered was that a purine, which is longer, should actually be paired with a pyrimidine, which is relatively what? Shorter, like that. So you've got one nitrogenous base. Let's say nitrogenous base, I'm using NB to be nitrogenous base. I'm using NB to be nitrogenous base. Okay, so you have one nitrogenous base here, and you've got another nitrogenous base on the other end. So this nitrogenous base is actually longer. So that is your purine. So you should have a purine. Then on the other side, you should have a pyrimidine. Okay. So a purine should actually be bonded to a pyrimidine. A purine should be bonded to a pyrimidine. So what we mean is we should understand what we call base pairing. Base pairing in DNA, okay? So purines and pyrimidines go together. So the way they move is that cytosine bonds with guanine. Then adenine bonds with thymine, like that, okay? So cytosine bonds with guanine via a triple hydrogen bond. You guys did hydrogen bonds, I'm sure, okay, when you did water. So this is a triple hydrogen bond, triple H bond. Okay. Triple H bond, while adenine bonds with thymine via a double hydrogen bond. Via a double hydrogen bond. Are people able to get me? I can see some comments coming through that people are unable to get me. Can I get some feedback? Are you able to get me?
We are able to get you.
Oh, all right, thank you so much. Okay, so we have this combination. Cytosine bonds with guanine, adenine with thymine. Now, remember, this is in DNA. Okay, now in RNA, in a place where there is thymine, there is actually uracil. So adenine bonds with uracil in what? In RNA. In RNA, adenine bonds with uracil. While in DNA, adenine bonds with thymine. It is important. That is a serious MCQ question. On the MCQ questions, they like asking that. You should know that in RNA, adenine pairs with uracil. Okay. So in this structure here that we are drawing here, whenever you see adenine, on the other end, you should have thymine, and there's a double bond. Whenever you see guanine, G, you should have cytosine on the other end, like that, okay? So you have that combination. Cytosine, guanine, okay? So DNA is a double helix. Okay, it's a double helix, as you can see, and it has got nitrogenous bases. And you should understand that thymine bonds with adenine via a double hydrogen bond, and guanine with the cytosine via a triple hydrogen bond. Okay. So now we are going to, you're still able to see my screen, right? Are people able to see my screen even up to now? Can I have some feedback? Are you able to see my screen?
We're able to see your screen, sir.
Right, thank you so much. Okay, so now, when we look at the objectives, the first one is prove that DNA is capable of accurately reproducing itself. Then the second objective is to explain the semi-conservative mechanism of DNA replication with the reference to the experiments of Marcelson and Starr. Then the third objective is to list the enzymes and proteins that are involved in prokaryotic DNA replication and explain the role of each of them. Then explain the different stages of prokaryotic DNA replication. Then we should differentiate between synthesis of the leading strand and that of the lagging strand. Okay, so understanding what we call semi-conservative mechanism of DNA replication. What they mean by semi-conservative, semi means what? Half, okay? Semi-conservative, to conserve is to actually preserve the original, okay? To conserve is to preserve, okay? So that there is a part of the original in the new product that you have actually formed, okay? So if you are saying, imagine you're making new bread, okay? You've got one loaf of bread, a new loaf of bread that you've bought, okay? Then you only say use half of it. You only use half of it. Then the following day, you buy new bread and get the odd half of that bread and you mix with the new half of the bread that you've bought today. So that bread is going to be made up of what? Of the new half and the old half. What it simply means is that you have preserved the old half of that loaf, isn't it? Okay. In the formation of the bread or the new bread that you have today, you have combined the bread, the half of the loaf of the bread that you used yesterday and half of the loaf of the bread that you've bought today. So what you've done is you've conserved, okay? So it's the same concept with semi-conservative mechanism of DNA replication. It's the same concept of semi-conservative mechanism of DNA replication. What it means is that, let me go back to the camera here, okay? So semi-conservative So what you're actually forming is you're producing new DNA. You're producing new what? New DNA from the old. Now remember that DNA has got how many strands? It has got 2 strands, okay? Say this is our old DNA, and then we duplicate, okay? To duplicate is to make another copy, is to replicate. It's the same term, duplicate. So DNA duplication or DNA replication is the same thing. It simply means that make another copy, okay? So now what it means by semi-conservative is that when making another copy, say we've got our daughter DNA this side, okay? Now, what happens is, here you've got hydrogen bonds, okay? You've got hydrogen bonds between those nitrogenous bases that we have, guanine, cytosine, adenine, thymine, you've got hydrogen bonds, okay? When DNA is replicating, the strands separate. The strands separate. The strands separate. When the strands separate, you're going to have two separate strands like that, okay? Now, when they separate like that, this separated strand is actually going to have another new strand that is going to form next to it. And this one also have had another new strand forming to it. So at the end of the day, from one DNA molecule, you're going to get two daughter cells. You're going to get two daughter DNA molecules. So not cells, but molecules, okay, like that. So what it simply means is that in the daughter DNA molecules that have been formed, you're going to have the original strand. Remember, you have the original strand. Even there is that original strand. Now this original strand, that is the odd strand. The odd strand and the new strand. The odd strand and the new strand. So the daughter DNA that you're going to produce is actually going to have an odd strand and a new strand. It's going to have an odd strand and a new strand. And remember that we're talking about semi-conservative mechanism of DNA duplication. What has happened is, in this daughter DNA molecule, there's preservation of the odd strand. The odd strand is still there, yet there's a new strand. Okay, so this is what they mean by semi-conservative. Semi means half. Semi-conservative. So it is actually, we have actually conserved. We have actually conserved the what? The odd. The odd what? The odd strand has been conserved. Wow, we have formed a new strand in the formation of what? New DNA. Okay, so this is what they mean by semi-conservative what? Mechanism of DNA synthesis. Semi-conservative mechanism of DNA synthesis. Now, we're going to look at the actual mechanism of DNA replication. We're going to look at the actual mechanism of DNA replication. Okay. So when we come to 3.4, we appreciate something about DNA replication in prokaryotes. That DNA replication in prokaryotes is well represented by E. coli. By E. coli, the process involves the following steps. So you should understand the steps involved. So the first step is replication initiation. Okay, you can read that. Replication initiation or initiation step. The second step is DNA denaturation. DNA denaturation, that is melting or unzipping. Remember, the hydrogen bonds that are actually found in between the nitrogenous bases, cytosine, guanine, adenine, thymine, those, it's like that is a zip, you understand? So unzipping or melting would mean that is the process that is going to be involved in the separation of the two strands of DNA. In the separation of the two strands of DNA, when that is done, there's another process known as priming. Priming, then after that, elongation, DNA synthesis at what we call the growing fork, then separation of circular daughter molecules. Then finally, we have what we call proofreading. So let us go step by step. We begin with initiation of replication in E. coli. Okay. So the replication of DNA requires the removal of what we call the supercores and the attachment of enzymes and other proteins to the origin of replication on the DNA molecule. What you should understand is that DNA has got what we call the origin of replication. There's one point, or there's a point where DNA duplication is supposed to start from. Okay, there's that one point. Okay, so understanding that there's that one point and DNA is actually forming a double helix, it actually cores up, it forms the cores, okay? It cores up, and because it cores up, there's a possibility of formation of what you call supercores. So the supercores are supposed to be removed. So the supercores are removed by an enzyme called topoisomerase. Topoisomerase is the enzyme that removes the supercores by the attachment of a protein called DNAA. DNAA, that is how it is done. So this results in the formation of what we call the initiation complex, the point where initiation is going to start from. As you can see, the demonstration there of the supercode DNA, then topoisomerase enzyme comes to act on it, then it will uncoil that DNA. It will remove the supercores. After removing the supercores, then DNAA will be incorporated into this uncoiled DNA to maintain or prevent DNA from coiling back, from forming the supercores again, okay? Then we have what we call the initiation complex. We have what we call the initiation complex, okay? Now, let us look at double-stranded DNA denaturation. What do they mean by double-stranded DNA denaturation? So the process is also called DNA melting or unzipping. So when DNA binds to the double helix, the first separation of the two strands take place. Remember, DNA is double-stranded. So when DNA binds to the double helix, as we saw on the other demonstration, there is actually unzipping or separation of the two strands at that point. Now that process, of course, requires energy in form of ATP, okay? After which it is followed by binding of the enzyme known as what? DNA helicase. Now, you should understand that DNA helicase is an enzyme that is responsible for unzipping. It will separate the base pairs, that is all the nitrogenous bases that we have, the complementary base pair in the cytosine to guanine, adenine to thymine, those hydrogen bonds will be broken. Remember, unzipping is actually simply breaking down hydrogen bonds, not the covalent bonds. Remember, we are separating the two strands. We are separating the two strands. DNA is a double helix made up of two strands. So we are separating the two strands because the two strands are simply joined by hydrogen bonds. Okay, so the denaturation of double-stranded DNA is what we call, is in a specific direction known as the 5' to 3' direction. The 5' to 3' direction, then the single strand binding protein prevents the double helix from rejoining. The single-stranded binding protein prevents the double helix from rejoining, okay? So they are actually demonstrating what happens with helicase. So helicase is an enzyme, as you can see on the demonstration there. We've got our initiation complex, then helicase comes in, which needs ATP to remove the hydrogen bonds, and the two strands begin to form. So you can see DNA single strand separated there, and they've put an arrow indicating the direction of denaturation, okay. After that, then we've got formation of RNA primers, okay. So now this process is called priming. This process is called priming. It is well known that DNA polymerase cannot initiate. Now remember, DNA polymerase is an enzyme that is responsible for the synthesis of DNA, for the formation of new DNA, okay? Now this enzyme, because we've already formed an initiation complex and we've started separating the two strands, okay? After doing that, then another enzyme comes in. After helicase has worked, another enzyme comes in, which is our DNA polymerase. Now, our DNA polymerase cannot initiate a new DNA chain, but can only elongate a pre-existing DNA or RNA strand. It is important to understand that DNA polymerase only can elongate that. Now, we therefore, before the potential DNA is replicated, a short RNA molecule A short RNA molecule complementary to the DNA strand is actually synthesized by an enzyme known as RNA polymerase, just a short segment of RNA, single-stranded. Remember that RNA is single-stranded. An RNA strand is actually synthesized. This RNA molecule is called a primer. This RNA molecule is called a primer, and it attaches itself to the initiation complex. A primer is actually synthesized on each DNA strand in the 5' to 3' direction. Okay, so as you can see on the diagram on this Y-like shape, you can see the leading strand primer, okay, on the 3', 5', and you can see the lagging strand primers, okay. So you have priming on the leading and lagging DNA templates by the action of primase. Primase is the other name for RNA polymerase. Primase is the other name for RNA polymerase. Okay, now let us look at the actual DNA synthesis. That is addition of now the nucleotides at the growing fork by E. coli DNA, by E. coli DNA. Now remember that we said that DNA is a polymer, isn't it? A polymer of what? Nucleotides. A polymer of nucleotide, because that's our nucleic acids. So we need to start adding other nucleotides, okay? Complementary now to form another strand by semi-conservative mechanism, okay? So this is a polymerization reaction catalyzed by DNA polymerase, although the two strands of DNA double helix are antiparallel. Antiparallel meaning they face opposite direction. One strand is in the 3' to 5' arrangement, then the other one is in the 5' to 3'. Okay, so it is important to understand that. Therefore, each strand is elongated only in the 5' to 3' direction. So it is important. Another MCQ question or another question that may be asked in an exam is to understand the order of DNA replication. That elongation takes place in the 5' to 3' direction. Okay. So synthesis of a new DNA strand involves the addition of deoxyribonucleotides, triphosphates. So these are our which include deoxyribonucleotides, adenosine triphosphate, okay. Then we have deoxyriboguanosine triphosphate. Then we also have deoxythymidine triphosphate and deoxycytidine triphosphate. So all these are the different nucleotides that we actually need. Okay. So let's go to the formation of a leading strand. Formation of a leading strand synthesis. So now there are two strands during DNA synthesis. One strand is said to be the leading strand, while the other strand is said to be the lagging strand. Okay. So the leading strand, at the growing fork, at each growing fork, one DNA strand called the leading strand is synthesized continuously. Okay. Take note of the word continuously. The leading strand is synthesized continuously from the growing fork. From a single primer on the leading strand template, then the leading strand grows in the 5' to 3' direction, like the growing fork. So when they say growing fork, so this is what they call a growing fork, this structure forming a Y-like structure or kind of conformation. So that is what they call a growing fork. Okay. So it is important to take note that the chain grows in the 5' to 3' direction. Now, if you come back to the growing fork here, or to the replication fork, okay, you can see a 5 and a 3 at the end of these fork. of this replication fork. There's a 5' to 3' end and 3' to 5' end. So they are saying that elongation happens in the 5' to 3' direction. In the 5' to 3' direction. As you can see, the leading strand is arranged in the 5' to 3' direction there, as opposed to the lagging strand, which is arranged in the 3' to 5'. Okay. So now let us look at the lagging strand synthesis. Now, synthesis of the lagging strand. To lag, it simply means to delay. So one strand is synthesized at a very fast rate, or the other one delays a bit. Okay, so that's why you've got two strands: a leading strand and a lagging strand. Okay, so the formation or synthesis of the lagging strand. So synthesis of the lagging strand is actually more complicated. It is not easy. Okay, so because DNA polymerase can add nucleotides only to the 3' end of a primer. Okay, that's the reason why it is difficult. Okay, DNA polymerase can add nucleotides only to the 3' end of a primer or growing DNA strand. Movement of the growing fork exposes the lagging strand template on which short RNA primers are copied. Each of these primers is then elongated by addition of deoxyribonucleotides, okay, to the 3' end. So in E. coli, this reaction is catalyzed by DNA polymerase 3. Remember, there's DNA polymerase 1, 2, 3. So 3 is the enzyme we're talking about now, DNA polymerase 3. The resulting short fragments containing RNA are covalently linked to DNA, which are covalently linked to DNA are called Okazaki fragments. The short fragments containing RNA, which is covalently linked to DNA, are called Okazaki fragments. In bacteria and bacteriophage, Okazaki fragments contain about 1,000 to 2,000 nucleotides. The overall direction of growth of the lagging strand is from its 3' end to 5' end, complementary to the polarity of its template, but opposite to the direction of nucleotide addition by DNA polymerase. Now remember, we had DNA polymerase 3, which is the one which is adding the nucleotides and making the DNA chain to grow. We've got DNA polymerase 1, which is primarily involved in removing RNA primers from the Okazaki fragments and filling the resultant gaps using its 5' to 3' polymerization activity. DNA ligase then comes to complement or to join those Okazaki fragments. DNA polymerase 2 functions in the inducible SOS response and also fills gaps and appears to facilitate DNA synthesis directed by damaged templates. DNA polymerase 2 resembles DNA polymerase 1 in its activity, but is a DNA repair enzyme, bringing about the growth in 5' to 3' direction. So what you should understand here is that DNA polymerase type 2 is actually a repair enzyme. It is a repair enzyme. Okay, it resembles DNA polymerase 1 in its activity. Okay, so let us look at the replication fork. You've got a leading strand, you've seen the arrow there. Okay, 3' to 5' direction. Okay, then the lagging strand, 3' to 5'. We've got the Okazaki fragments. Then we've got direction of fork movement, direction of fork movement. Then we've got the parental DNA duplex, then the daughter duplex. Okay. So that this diagram is showing the replication fork. Then there's what you call proofreading by DNA polymerase. Proofreading by DNA polymerase. That is to check that there are no errors during DNA replication. Remember, errors can occur during DNA replication where you've got base mismatch. You know that guanine is supposed to bond with cytosine, You can have errors where you have guanine bonding with what? Adenine. So that is an error that is supposed to be corrected. So you've got enzymes that are actually dedicated for proofreading and correction of these errors, okay? So occasionally a wrong base is inserted into DNA synthesis, then this can actually be corrected by proofreading function of all bacterial polymerases. So all the polymerases, that is DNA polymerase 1, 2, 3, they can actually perform what? Proofreading. They can perform proofreading. Then during replication, separation of secular daughter molecules, during DNA replication, the parent strands remain intact and retain their super helicity, okay, from the word helical or helix. This poses steric and topological constraints on the completion of the replication of a circular DNA molecule as the two growing forks approaches each other. In E. coli, decatenation of the two daughter cells is catalyzed by topoisomerase or DNA gyrase. For this helps separate the daughter DNA molecules from each other. Remember, this topoisomerase is just preventing the two circular DNAs from forming cores on each other, okay, so that they are separated, they are independent. Okay, so now what is important to take note of, again, is the enzyme, the specific enzyme involved in DNA replication and its function. So even when they ask in MCQ questions, they would ask you MCQ questions on replication based on the different enzymes and proteins and what their function is. So this table 3.1, is very important for you guys to understand. So the first one is just in summary, DNA protein. This one binds to form the initiation complex. It binds to form the initiation complex. Then we have topoisomerase. Topoisomerase, this one removes cores and supercores from DNA. Okay. Then you have DNA polymerase 1. DNA polymerase 1. Then you should know the other name for topoisomerase. That is DNA gyrase. DNA gyrase is the other name for topoisomerase. Its function is to remove the supercores and the cores from DNA. Then you've got DNA polymerase 1, 2, and 3. DNA polymerase 1 is the one that removes the primers from the new DNA strands. Then DNA polymerase 2, this one is involved in the repairs mistakes during DNA polymerization, and also fills in the gaps left after the removal of primers from the lagging strand. DNA polymerase 3 is the one responsible for DNA polymerization, for formation of that primer. Okay, then helicase is involved in DNA denaturation, that is removal or breakdown of those hydrogen bonds between the nitrogenous bases, guanine, cytosine, adenine, and thymine. Then we have another enzyme known as primase enzyme. Primase enzyme is used in the synthesis of short RNA primers on template DNA. Okay. Primase is also known as what? RNA polymerase. You should take note of that. Primase is also known as RNA polymerase. Then you've got ligase enzyme, which is responsible for joining DNA nucleotides through covalent bonds. Joining DNA nucleotides through covalent bonds, as it helps in the synthesis of the lagging strands. Then you've got the single strand binding protein. This one prevents single-stranded DNA from forming double strands. Okay, so basically that was DNA replication. So the other thing that you should understand is that how do you move? So from DNA replication, you've got the duplication or formation of another copy of DNA. Then when that has actually been formed, the next step that follows replication is what you call transcription, which is formation of messenger RNA from DNA. So that will be the next topic. Then from transcription, you go to translation, that is formation of proteins from messenger RNA. That is another topic. So that is what you call central dogma of molecular biology. Central dogma of molecular biology, that DNA undergoes replication, and from replication it will undergo transcription, where messenger RNA will be formed from DNA, then later on, proteins will be formed by translation. That is what you call central dogma of molecular biology. So that was replication. So unless there's a question, now that is where a replication ends. Do I have a question? Otherwise, if there's no question, our time is running out. We are actually done. We are actually done with the session. Hope it was clear. Silence means clear.
Yes, it was.
Oh, okay, excellent. Okay, so we can call it a day, then we'll arrange for another session next time. Okay.